give us the time. We could have put this a little bit earlier, but this is our usual time, so it's good to keep it that way.
Yeah, pleasure. Thank you so much for inviting.
Yeah, maybe 1 more minute, and then I promise to be brief in my introduction.
All right, should I try and share my slides and make sure everything's
Yeah,
working?
I think that's a great idea. Why don't you go ahead and do that? So at least we use the minutes this way and be sure. Okay, so we see your slides.
Perfect.
Put it in presentation mode and make sure the back and forth also works.
Come on. Do you have a presentation?
Yes, but we see your notes. So
okay. There we go.
Okay, perfect.
Ah.
Do you like to check the back and forth works? Okay, perfect.
It work.
Awesome.
Let me just actually do it the other way around. I'm gonna put this down here. I can't see you anyway, which is horrible.
I think it's fine. Yeah, it's making sure the slides are.
Which one do you see now?
The good one.
That was the good one.
No, but now we see the notes. Okay.
Now you've got it the right one, yeah?
The right one, yes.
How can I? Okay, let me see if it's just annoying because it's the big one that's on my screen. I'm trying to see if I can. Oh, wow. I'll go with it as it is.
Okay, so if you don't mind, maybe I will go ahead and introduce you. Good afternoon, everyone. My name is Golnaz Vahedi, and I'm a faculty member in the Department of Genetics. It's an absolute pleasure to introduce our today's speaker for the Penn Bioinformatics Forum, Dr. Gosia Trynka. Dr. Trynka is a group leader at the Wellcome Sanger Institute in Cambridge, UK. And she's also the director of experimental science at Open Targets in Cambridge, UK. Dr. Gosia is from Poland, and she received her PhD in the Netherlands, and then followed her studies as a postdoctoral fellow with Shomoya Ray Chowdhury and Robert Plenge at the Harvard Medical School. I think I know her because of her absolutely fantastic work that she did in Shomo's lab on linking the genetic variants associated with autoimmune disorders, and really wonderful studies that she published with Shomo and Robert Plenge. And as a group leader, she has contributed a lot to better understanding of how variants affect cis-regulatory elements with a particular focus in the immune system. So without further ado, I would like to all of you to join me in welcoming Dr. Trynka, and we look forward to hearing from you. And please type your questions in the chat, and we look forward to have a really great and interacting seminar. Thank you.
Thank you so much, and thank you very much for your kind introduction, and also thank you very much for the invitation. And it's been fantastic to have this remote interactions and get to know what are you working on, and then I've had quite a few productive discussions, so thanks so much. So what I thought I'll do today is present you with pretty much focus on two studies that we've been running in the group in the past few years that really focus on understanding or trying to understand how GWAS variants associated to immune diseases, which is our primary focus in the group, how do they manifest and lead to the diseases? And so for that, we're combining a range of different immunology and genomic approaches to try and see what's the impact of genetic variation on the immune system. And so we think a lot about common immune diseases, and these are really a collection of something around 80 different phenotypes. And they can have different manifestation, and collectively they affect about 10% of general population. So they can have very specific manifestation targeting specific organs of our body, such as, for example, small intestine in celiac disease or the synovial tissue in rheumatoid arthritis. But what I think is really fascinating is that we know that there's a genetic overlap between immune diseases, and we know that one shared component across immune diseases is the dysregulated immune system. And as I mentioned, this is really the at the heart of what we're doing in my group, trying to understand how this common immune disease variants dysregulate our immune system to predispose to disease development. And this is a schematic of a roadmap that we take when we think about the how to functionally follow up GWAS-associated signals. And the first question that we want to answer is, which are the disease-relevant cell types? And it is an important question to answer because, as you'll know, majority of the GWAS-associated variants are non-coding, and therefore they are implicated to play a role in gene expression regulation. And gene expression regulation can be cell type specific. Therefore, follow-up functional studies need to also be performed in the most informative, most relevant cell types. Second of all, once we have that cell type of interest, then we go on to further investigate what are the disease causal genes, what are the exact regulatory variants, and also what are the exact cellular functions that are dysregulated by these associated variants. And in the context of immune diseases, identifying which are the disease-relevant cell types actually is somewhat challenging. And that's partly because the immune system comprises really a large range of different immune cell types. And those cell types also, the way they function through interactions, through communications between different cells. And that sort of triggers different cell states among cells. So it's not only that we don't know exactly which are the relevant cell types, it's also the context in of the cell types that is important for understanding gene expression regulation. And one way through which we've been, we and many others in the field have been thinking about connecting GWAS variants or using the GWAS associated signals to connect to the disease relevant cell types is through integration of the associated variants with chromatin activity profiles. And such chromatin activity profiles can comprise chromatin marks that highlight active enhancers and promoters, such as H3K27 acetylation, or can use open chromatin regions. And the different chromatin marks sort of have different sensitivity for this approach, so I'm not going to go too much into details on that. But the general concept is that we can take these associated variants, and then if we have a map of different tissues that have been assayed across, that have been assayed with chromatin marks, then we can ask a question: are there specific overlaps between the disease-associated variants and peaks that are specifically present in some cell types and absent in the others? So in this schematic picture here, Each dot would represent a GWAS-associated variant. And the question here is, if we look collectively at all of those variants, do we see that some of those variants are overlapping with peaks present in one cell type more than in the others? And in this case, we would see that 3 of the variants are hitting this 2 peaks that are really tall, indicating that this region of the chromatin is really active in the bone. And that would sort of implicate that there's a strong enrichment for disease variants in this particular tissue, indicating that this could be a relevant cell type to follow up further functionally. And so in using this approach, we have previously published different methods how one can integrate this disease-associated variants with chromatin marks. And this is particularly here I'm referring to the work that I worked on in Shoma's group in Boston during my postdoc. But the point I want to make here is that when we applied these methods to data sets that were emerging back in 2014-15, which included roadmap epigenetics data sets, the ENCODE data set, and we applied this across a range of different immune diseases, what we saw was that across different traits, there was strong enrichment of associated variants in different CD4 T cell subsets. And that was really interesting. I think it was particularly interesting because it sort of met with what we have known from immune studies that show that the CD4 T cells play an important role in many of the immune traits. But I think it was really, to us, it was really interesting because we could clearly demonstrate that the genetics is also harboring the same signal. So it pointed towards CD4 T cells. And the second step of that is understanding which cellular functions are really dysregulated by the variants in the CD4 T cells. And that's something that I want to talk a little bit more about today through the approaches that we've been using in my group. So just before we go on to the experiments, for those of you who are not immunologists, let me briefly explain what T cells do. And for those of you who are immunologists, I apologize for simplifying things quite a lot. But in a nutshell, CD4 T cells play a really critical role in orchestrating the response of the immune system. So in our body, when there's something foreign, when there's an antigen, it gets picked up by the antigen presenting cell. And that cell presents the antigen to the CD4 T cell. And in the presence of right signals, CD4 T cell will undergo activation. And upon activation, it will then go on to communicate with the rest of the immune cells to tell the immune system how to further proceed with the immune response. And that is dictated by the presence of different cytokines in the environment. And those cytokines can, for example, be secreted by the antigen presenting cell. So depending what's present in the environment, the T cell will acquire different effector functions and will become T helper cells. And then depending on which effector function it acquires, it will subsequently exert different functions. So, those functions can be manifested by secretion of other cytokines, but also T cells then start to express different ligands on the surface, and through those ligands and receptors, they can directly interact with other cells. And final part of this process, of course, is one thing is getting the immune system alert that there's something in the body that needs to be tackled, but there also needs to be a braking mechanism to this, or else the immune system would go on activated and would really escalate the inflammation. And that braking mechanism is delivered by regulatory T cells, which are a subset of CD4 T cells, and they provide a suppression which can be manifested either directly by, for example, expressing certain molecules through which regulatory T cells can interact with antigen-presenting cells, and also secreting different cytokines. And so, keeping in mind this circuitry, what I'm gonna do next is mostly focus on the activation component and the cytokines that polarize cells to different phenotypes. And if I have time, I'll try and talk a little bit also about the regulatory T cells and how we, and some of the approaches we've used to study regulatory T cells. But this is the circuitry that we decided to focus on to get better insights into what disease-associated variants do in T cells. So the first question we've asked was, which are the disease most relevant cell states that are induced upon cytokine exposure? And this project has been initiated and funded through Open Targets Consortium, which is a public-private partnership at the Wellcome Genome Campus that brings together the Sanger Institute, EBI, together with industry partners who currently include GSK, BMS, and Sanofi. And the overarching goal of Open Targets is to leverage genetics to better inform target identification and target prioritization. So with that, what we did is we proposed and executed this project, which was led by Blagoje Šoškić, who's a postdoc in the group and a really exceptionally talented postdoc. Together with Eddie Cano-Games, who's a PhD student in the group, also a fantastic scientist and really talented. And together, these guys have been really driving these efforts. And what we set out to do in this project was to isolate monocytes, which you can think of as the antigen-presenting cell that I was referring to earlier on. And we isolated CD4 T cells, and in particular, we isolated the two main cell types of CD4s, or subsets of CD4 T cells, the naive CD4 T cells, which haven't encountered activation in the body yet, and the memory cells that have been previously activated, so they somehow hold that memory of previously having seen the antigen. And then we took those cell types, and from monocytes we derived macrophages, and we stimulated all the cells in the presence of different cytokine cocktails. And we've used cocktails that would polarize T cells towards the major T helper subtypes, but also we've used some cytokines that have been previously implicated to be important for in immune diseases and in inflammation. And we've used slightly different set of cytokines for macrophages to make it relevant, of course, for the cell type. So upon stimulation in the presence of the cytokines, we then profiled chromatin activity at two different time points, and we have chosen different time points for different cell types to make them relevant. So for macrophages, because they are the innate arm of the immune system, so they are designed to rapidly respond, we profiled chromatin activity at 6 hours and at 24 hours. But for T cells, we've chosen 16 hours and 5 days, which would represent early and late activation time points corresponding to for 16 hours corresponding to cells just prior the first cell division. And at 5 days, it's more of a time point where cells should acquire that T helper phenotype. So they kind of matured. And we've used 3 healthy individuals as replicates, and altogether that resulted in a data set of 55 different cell states. And we've used ataxic and H3K27 acetylation to profile their chromatin activity. So the first thing that we notice, which was very much in line with what others have reported, is that upon activation, chromatin landscape undergoes major changes, and there's a lot of remodeling, and there's particularly a lot of regions that coming up as new peaks are arising upon activation. And that's present both in naive memory T cells as well as macrophages. And the second question we looked at with this data set was how many of those peaks that are induced was the word I was looking for, how many of those peaks that are induced upon activation are cell state specific. And actually, to our surprise, we saw that not very many. So the vast majority of the peaks that are induced upon activation are shared between 20 to 40 cell states of the 55 cell states that we're capturing here. And only really a very small proportion of those peaks is more context specific. That wasn't exactly what we were expecting to see. But the other thing that we noticed was that although there was a lot of sharing in those peak regions, there was a variable height of those peaks, a variable activity of those peaks across different cell states. And that's represented here by the coefficient of variation of read counts within each individual induced regions. And you can see that that variability is greater in H3K27 acetylation compared to ataxic. And from now on, majority of the analysis I'm presenting is actually focusing on K27 acetylation because we find it more informative for this analysis. But ataxic is suitable as well, it's just a little bit less powered. So this observation of this substantial sharing of peaks across different cell states had direct implications on what we were planning on doing. And what we wanted to do is really use one of the methods that I previously I developed in my postdoc in Sharma's group, and apply it to this data set. And what those methods do in general is that they look for this binary overlap between associated variants and the peaks. So they rely on the fact that you see a peak in one cell type and that peak is not present in other cell types. And that wasn't what we were dealing with. The situation that we saw in our data set was more of a quantitative differences between the peaks across different cell states. So Blagoje, who's an immunologist by background, who when he joined the group, he looked at this and he decided that my methods were completely useless. And so he decided he would develop his own methods, his own method that would specifically address this problem. And that's exactly what he did. He worked hard, picked up quite a lot of coding, and developed his own method. And in just very briefly, the way this method works is actually that we're looking at each individual peak, and we're scoring how cell type specific individual peak regions are for each one of the cell states in our data set. And then we rank these peaks, we rank the specificity of those peaks, and then we ask how we overlap the peaks with disease-associated variants, and then we essentially sum up the ranking of those peaks. And we have a permutation method, as well as we fit in a distribution to assess how significant the observed overlap is compared to what you would expect by chance. And the method is called CHEERS, that stands for Chromatin Element Enrichment Ranking Specificity, which is very difficult to find if you try and Google CHEERS, but it's also present. You can also find it on our GitHub. So we first of all applied this method to blueprint dataset to just see how it performs when you have a diverse set of different cell types. And we were very pleased to see that it was really recapitulating what we would expect to see. And here I'm showing you how the types of enrichment amongst across different immune cell types for different immune traits. And what you can see is that, for example, we can see that there are diseases that seem to have very strong T cell components. So for example, allergies and asthma, they have a very strong enrichment in both CD8s and CD4 T cells. While for the other diseases such as lupus or systemic sclerosis, we don't pick up much enrichment in T cells, but we see that there's a very strong B cell component. And for Andrew, unfortunately, Blueprint didn't have T follicular helper cells, so we couldn't test that in here, but I would be really keen to see how that works, because I think that we would observe some enrichment. So then we applied it to our data set. And here's an example for IBD demonstrating that we can pick up a signal in individual cytokine-induced cell states. So in this case, this is for late activation of naive T cells. And this is with cytokine cocktail polarizing towards Th1 phenotype. So the way to read through these plots is you have the significance of enrichment, and then different shades of colors. If they lighter shade, they indicate early time points, and the darker shade indicate late time points. Purple are memory T cells, green naive T cells, and the yellow is the macrophages. So in this case, it was really encouraging that we could see across IBD and the different subphenotypes of IBD, as well as celiac disease. We clearly saw that there was a strong enrichment in Th1. And that was really encouraging because there's really a lot of evidence showing Th1 cells playing a role in lamina propria of patients with these diseases. But more globally, what was really interesting to us was that we didn't actually see those individual states coming up. What we saw was the predominant signal was captured by T cell activation, and that was really striking. You can't very well see resting cells here, but the first bar in here in purple or in the darker purple, you see there's really not much enrichment at all. But what we saw altogether was this strong enrichment for disease variants in early activation time points, especially in memory T cells. And how does it look like when looking at individual trait? So this is the distribution of specificity scores for variants associated with IBD. So we've got variants, different loci on the y-axis, and here you have the distribution of the different immune cell states in our data set, and you see that there are those different patterns. There are loci that are very specific to late activation time points, and then there are loci that are specific to early activation. And what driving the enrichment is really just accumulation of a little bit more of the variants that are pointing towards early activation of memory T cells. And when we take genes nearby peaks that are driving this enrichment, enrichment, and we run it through pathway analysis, not surprisingly, we see that exactly we're picking up lymphocyte activation, T cell activation pathways, and so on. And finally, how it looks like when you zoom in at a individual locus. So here's an example of a variants associated with allergies, here marked in yellow. And you see that they are overlapping with peaks that are very specific, that are very, with a region that is particularly active in early activation of memory T cells without any cytokines. This is actually just beats for T cell activation. And then other examples, such as for rheumatoid arthritis variants, can include variants that are overlapping with these peaks that are present in early activation, both in naive T cells, in memory T cells, but you don't see much chromatin activity here in the resting state or in the late activation time points. So with that, just to summarize this part of my talk, we generated this data set of chromatin where we provide chromatin landscape across 55 cytokine-induced immune cell states. We developed a new method called CHEERS that takes into account quantitative changes in chromatin activity to perform SNP enrichment. And that pointed towards the importance of early T cell activation, particularly in memory T cells. And this work has been driven by Gosia and Eddie. And so we then thought, all right, so we've got this indication that T cell activation is really important for immune cells. So how can we follow this up, and how can we better understand what is the genetic control or regulation of the T cell activation? And so for that, what Eddie and Gosia did was to generate a single cell eQTL data set from healthy individuals where we performed a time course of T cell activation. So we've recruited close to 120 individuals, and from those individuals we isolated naive and memory T cells, which we then stimulated with beats, so there's no more cytokines in this part. And then we profiled gene expression with single cell with 10x. We profiled at 4 different time points. So we've used the resting time point, and then we used 16 hours, which is just before, again, cells undergo first cell division, 40 hours, which is at the more or less at the time or just after cells have undergone the first division, and then again 5 days when T cells acquire maturity. And one of the motivations why we wanted to use single cell sequencing in this case was the fact that we've in another study, Eddie and Gosia used single cell profiling to look at T cells, and what we observed was that transcriptionally T cells actually don't form this separation between naive and memory T cells. They actually form like a continuum. And particularly within memory T cells, it's known that there are different subsets of memory T cells. And you can see that with transcriptionally, you get to see that there are central memory T cells, effector memory T cells, TEMRAs, and regulatory T cells. And so there is this heterogeneity, particularly within the memory T cell compartment. And again, the T cells don't really, transcriptionally, they don't cluster into very well-defined clusters. They form this continuum. And so we wanted to capture that information, and that's why we decided to go for a single cell transcriptional profiling. So with this approach, what we observed was we've generated a data set of actually over 700,000 single cells, but after QC, we ended up with over 600,000 650,000 single cells that contributed to the final analysis. And first of all, what you can see is that we can clearly see the separation between memory T cells that sort of form in the inner part of the UMAP and the naive T cells that are in blue. And again, you can also appreciate that, just like I was saying on the previous slide, you don't see very clear separation. You see that the cells are forming this continuum. Second of all, we can look at which time points contribute to the different parts of the UMAP. And what we see is that we have this clear cluster of resting cells that are separated a little bit from all of the activated cells. And then we have the 16-hour stimulated cells, 40-hour stimulated cells, and 5-day stimulated cells. And what was really interesting, and something that we wouldn't have identified with bulk RNA-seq, was that we also observed there was this cluster of lowly active cells that were falling in between the 16-hour stimulated and resting. And really, there were cells in this compartment that were contributing from all of the different time points, but a lot of them were coming from 16 hours and 40 hours stimulated cells. And it was it included both naive and memory T cells. We then went on to perform EQTL mapping. And for that, first of all, what we did is we just created a simple pseudobulk to understand how many EQTLs we can map when we just look at the pseudobulk of resting cells, 16 hours, 40 hours, and 5 days. And you see that with time, we are picking up more and more EQTLs. And importantly, you start to see these patterns, that there are differences between EQTLs in naive T cells compared to memory cells. So in this case, for IL-12 RB1, you see that in naive T cells, we don't see any EQTL effect, but there's a correlation in memory T cells. And then we're also starting to see that you can pick up those effects that are not only cell type specific, but they are also a time point specific. So here, for example, GFPT1 is an EQTL, is not an EQTL in naive cells, but it is an EQTL in memory cells at 16 hours stimulation. But of course, because we've got the single cell resolution, we also wanted to look at what's the exactly heterogeneity of T cells upon activation. And we were able to identify altogether 38 distinct clusters throughout the activation time course. And some of those clusters were stable across all of the time points. So for example, central memory, effector memory, and TEMRAS, we could see those clusters coming up in each individual time point. But then also with activation, we saw that there were some clusters that were more sort of transient. So for instance, we saw that in lowly active and from there on throughout activation, but not in resting cells, we could see a cluster of T cells producing interferon genes. Also, we observe a cluster of cells produced that were expressing NF-kappa B genes, and that was present at 16 and 40 hours. And then we also observe a cluster of mitotic cells at 40 hours and 5 days, which sort of indicates cells are starting to divide, and we were picking up some cycling cells. And of course, with that, we can now ask a question. So can we pick up any EQTLs that are specific to individual time points and subpopulations? And indeed, that's the case. With single cell resolution, we can identify effects that we would have otherwise missed if we had bulk RNA sequencing. So here's an example of TYK2. And when we use the pseudobulk of all memory cells, you see that this is looking specifically at 16 hours, you don't see any effect. But then as we start looking into individual clusters within this time point, we see that actually TYK2 is an EQTL in effector memory cells, but not in central memory cells. And so this effect gets sort of diluted when you put all the cells together. And it is interesting because ultimately what we wanted to do is perform this colocalization analysis between GWAS-associated signals and EQTLs. And what you do in the colocalization analysis is that you're asking a question if the patterns of association between EQTLs and GWAS are shared. And when we apply that in our data sets, in our data set, we actually observe that for instances like TYK2, we do see that there's a colocalization with IBD associated variants and this EQTL effect in effector in effector memory T cells, which is really interesting because TYK2 has also a really interesting, has been shown to play a role in IBD through coding variants. So this sort of adds in an extra layer of information that there's not only the coding variants that can regulate T cell function, but also there is some T cell function regulation through the levels of expression of TYK2, and very specifically in early activation of T effector memory cells. So as I said, we performed this colocalization across a range of different immune diseases. We've used 14 traits for that, for which we were able to access summary statistics. And we observed altogether over 300 colocalization events. Now, one caveat with this type of analysis is that although we have this ability to map a lot of different cell clusters, it is really difficult to have confidence in calling EQTL effects in clusters that are comprised of small number of cells, because you're just unable to reliably call mean gene expression, and there's just too much noise. So effectively, for a lot of the, some of the clusters that are small and present across different time points, we actually are only able to map a handful of EQTL. So it's not really fair to look at those clusters, and you can see those indication of the number of EQTL genes that we're identifying per population in this bar plot here. So as the clusters become bigger and more reliable, then we start to map more and more EQTLs. Of course, there's a saturation point. At some point, you've got enough power, and then you start to see EQTL effects that are actually biologically meaningful. So, you know, upon activation, for example, you start to see more of those EQTLs. And then you can also see that we're mapping this colocalization effects among GWAS loci, and you see what's the number of significant loci reported for a GWAS. And there is a little bit of correlation with how powered you are for a given GWAS, but overall, actually, there is quite a variability. So you pick up more colocalizations with some traits over the other, even though some of those traits have quite a lot of reported associations. So for example, SLE here has quite a lot of GWAS loci that are reported that we're testing, but overall you're not seeing that many genes colocalizing, which is in line with our earlier observation showing that variants associated with SLE are more enriched in B cells rather than in T cells. Okay, let me move on. Right. So another type of analysis that we did with this is that we looked at whether, we looked at gene expression of activation markers. And what we saw was that the cells were you could see that there was a continuous change of gene expression throughout the activation time. So here's the activation time, and here's the changes in gene expression. Some of the markers are very well known expression markers. So for example, IL-7 receptor, which is known to be highly expressed in resting cells, and the expression goes down upon activation. We clearly see that. Then we've got genes such as CD69, which is a marker of early activation, and it exactly comes up at around 1640 hours of activation. Then we've got IL-2 receptor A, which comes up later on in the activation. So this kind of continuum of gene expression activation made us think that perhaps we could maybe use the gene expression more as a continuous, use the time of activation as a continuous variable to map EQTLs. So for that, what we did is we applied a pseudo time to our activation to infer a trajectory. And what you see is that the trajectory starts here, and then it goes around exactly as I showed you before, that this is from resting state throughout early activation all the way to late activation. And then we can take the pseudo time values and we can order cells based on the pseudo time values, and now check again what's the expression of the known activation markers. And when we did that, we observed encouragingly exactly what we wanted to see, which I referred to earlier in my previous slide, so that, for example, IL-7 receptor expression goes down upon activation, IL-2 receptor A activation goes up in early activation time points, and then it plateaus. And then CD69 is a marker of early activation, so it sort of goes up and then goes down. And with that, we then thought, okay, so let's use this activation pseudo time to perform dynamic EQTL mapping. And what I mean by that is that we ordered the cells according to pseudo time, and then we bin cells into 10 different bins. And then we mapped EQTLs in each individual bin. And what we're looking for is these effects of EQTLs that are sort of attenuated throughout the time of activation. So in this case, you don't see any effect at early time points of activation, but as the cell progresses through the activation with different genotypes, you start to see that there's an effect on gene expression. So we've done that both in a linear manner as well as in a quadratic, using a quadratic model, so that we can identify those relationships that are linear, but also those events that you see that there's a strong effect in the early time points of activation, but then the effects are gone as you progress throughout the activation, and in late time points of activation, you don't get to see any effects. And with that, we observed that about a third of all of the EQTLs in our data set actually had this dynamic regulatory nature during T-cell activation. And then, of course, again, what we're doing now is using this dynamic EQTLs to perform colocalization with GWAS-associated variants. And in this case, I'm showing you an example of CTLA4, which indeed expresses this dynamic effect. It has a EQTL effect in T memory cells in early activation, and it is colocalizing with type 1 diabetes association. And the final point is that when we look at the immune disease colocalizations and compare how many of the colocalization effects are with dynamic EQTLs compared to non-dynamic EQTLs, we do see that there is indeed enrichment of effects. There's enrichment of colocalizations with dynamic EQTLs over the EQTLs that don't have that dynamic effect, both in memory and naive T cells. All right, so with that, in summary, we have generated what we think is, what I'm aware of is the first single cell EQTL map of T cell activation. And with that, we've identified 38 distinct clusters from across T cell activation time course. I showed you examples of this cluster and time point specific EQTLs that would have been missed if you had performed bulk RNA sequencing. A third of all the EQTLs that we see express this dynamic effects. With that also we see that there's enrichment of disease associated variants in dynamic EQTLs. And that is not so good news to those of us who are trying to understand how those disease-associated variants function, because it just demonstrates, I think, how plastic immune cells are and how dynamic gene expression is, and that it's going to be it's going to keep us busy for a little while to really try and figure out the functional effects of GWAS-associated variants. So I'm gonna skip through my Treg story. I'm just gonna close with this one sentence, with this one last slide, which I think really, I'm really passionate about combining different approaches for us to get us closer to understanding how GWAS variants function and to accelerate that knowledge to drug development. And I think the biology is so complex, we really will need a whole army of different approaches and expertise and collaborations between immunologists, cell biologists, geneticists to really figure out at least some of the genetic function of some of the genetic variants. And with that, I'd like to acknowledge my team. There's some of them, not all of them, but especially I mentioned the work that was led by Blagoje Šoškić and Eddie Canogames, who are absolute superheroes. And this project has been funded by Open Targets, and we've really enjoyed a lot of interactions, great interactions with our partners, especially in GSK and in Biogen. And there's some of my wonderful immunology collaborators who help in general in steering us in the right directions in our research. Thank you very much for listening, and I'm happy to take any questions.
Wonderful, really fascinating talk. Thank you so much for sharing. You have a lot of questions, so I think I'm gonna go through them, and some of them are related, so hopefully maybe we can address as many during the next few minutes. Catherine Sustak is asking, the single cell EQTL are really exciting. How robust is the data set given the sparsity of the data? Can you replicate all bulk EQTL sets? In addition, have you compared to single cell EQTL data calling to bulk deconvolution analysis?
So I'll start with the convolution because we haven't done it. But also I think where you might struggle with deconvolution is that, at least when I talk to, some of people I know who think a little bit more about the deconvolution than I do, is that deconvolution works well when you have very different, when you're deconvoluting very different cell populations, right? So if you have PBMC RNA-seq, and then you're asking a question, how much of that signal, a QTL signal, is accounted for by monocytes and T cells and B cells, which are very different cell types, then you have the power to do that. It doesn't work that well when you're trying to even say tease out Tregs out of the CD4 T cells. So we haven't done it, but my gut feeling would be that it probably wouldn't work too well. But if somebody wants to have a look at it, I'm happy for them to collaborate with us and give it a go. There was another part too. There was a lot at the beginning.
Yeah, maybe can I ask you a clarification question about maybe this question? And so you do genotype your individuals separately, correct?
Yes.
Got it. So then maybe the question here is, can you replicate all bulk EQTL sets?
Yeah, I don't know. We haven't looked into that properly. The issue there is that you always struggle with, like, you know, depth of sequencing. So we know from our other studies where we like look at comparison between naive and Tregs from bulk RNA sequencing, it becomes challenging because you have a lot of overlap, but because you've done deeper sequencing, for example, you have more power to detect those EQTL. So it becomes then difficult. That's why what we wanted to do in our study is use our data as a pseudobulk and then ask the question, how much do you miss when you have that pseudobulk within the same data set? So then at least the sequencing coverage is kind of similar.
Thank you. A quick clarification question from Sarah from the NIH. She's wondering where the cells come from. Did you obtain them directly from human tissues or blood?
Yeah, so they come from healthy volunteers. We get them through our collaborators at GSK, actually.
Excellent. Jen is asking, the pair population EQTL mapping is very interesting. I would like to ask Dr. Trynka, how you deal with LD structures when you do EQTL mapping?
How do we deal with it? That's a very technical question, and this is a very embarrassing PI answer, which is I have little idea because they're actual human beings who are doing the work, and it's not me. We run, I trust, what do? We run TensorQTL for this, and it gives you an index variant, and then we go with that index variant, and we just sort of use the standard framework. I'm not sure. Is the question about whether we do any conditional analysis or is it more if we're thinking about fine mapping those signals?
Yeah, I think maybe he can follow up with you and we can go through some of the other questions.
Yeah, and feel free to send me an email also, and I can point you to Eddie and Bragaya, who will give you a much more comprehensive answer.
Sounds great. Van is asking you, giving you a lot of praise, amazing talk. That lowly activated cluster caught my eye. I've only seen stem cell-like memory CD8 T cells due to recent interest and publication. Does T stem cell memory phenotype exist for CD4, and does it offer similar functionality and therapeutic potential?
I don't know. We don't have that. Well, we've not looked at it specifically in our data set. I agree that cluster is super interesting, and there's so many questions I've got around that cluster, and I think we don't have the data set to answer them, but I'm partly thinking, are there cells that are committing to activation and then not making it all the way through, and that's why they end up being in this slowly active state, and then they either reverse back or they die? or are we just capturing, there's like a distinct transcriptional state before cells fully go onto cell cycle and progress into proliferation. So I'd love to look a little bit more into that cluster and understand what's exactly comprising in that cell subset there. It's really interesting.
Okay, and maybe super quick, Bobak Faryabi wants to know, would you comment on whether and how looking at dynamics of chromatin accessibility could inform your single cell association study?
Yeah, so we haven't done any single cell ATAC-seq for this data set, sadly. We are currently looking into integrating some of those EQTL signals with the chromatin data sets that I presented in the first part of the talk. And I don't have the answer, well, I don't have a good answer to tell you what's the story behind it. It's something we're still working on to try and see if there's more information that we could bring together by merging the two data sets. They're done on different individuals and with slightly different protocols, but hopefully still we should be compatible enough to make some conclusions.
Wonderful. And the last question from KP Joy, EQTL analysis at the resolution of single cell is amazing. I wonder if the linear mix model was sufficiently stable to deal with the gene expression and stochasticity among multiple cells across donors in each cell states.
So, and again, Blagaj and Eddie will be much better placed to get into the type of conversation you would probably enjoy by asking this question. In this analysis, what we've done is essentially we're creating the pseudo-box for each of the clusters, and that's what we're taking into EQTL analysis. So we're not taking individual cells for mapping EQTLs. Yeah.
Well, so I would like to thank you for this very exciting seminar and everybody for participating and asking great questions. Thank you so much, and thanks again for visiting us virtually.
Thank you, pleasure. And if there's any more questions, feel free to send me an email. I'll be happy to answer.
Excellent. Thank you, everybody. Bye.
Thanks, bye.